What's going on, Science Stands? Mr. Walton here. Today we're going to talk about the cell membrane. And in our last lesson, we were talking about organelles inside the cell, the structures and functions of different organelles. Now, if you recall back to that last lesson, the cell itself is wrapped up in a giant membrane. And then a lot of the organelles inside are also wrapped up in a membrane as well. So this cell membrane is actually really critically important, and it's fundamental to our understanding of how the cell operates. A cell membrane helps the cell to maintain homeostasis. What that means is the cell is able to keep a balanced state. The cell wants to be able to keep a balanced state because it doesn't want to run out of nutrients or it doesn't want to build up too many wastes. Another name for the cell membrane is the plasma membrane. It consists of a phospholipid bilayer. What that means, bi means two. So there's two layers of phospholipids. Let's go ahead and draw a phospholipid. So I'm drawing a phosphate head and two lipid tails on them. This phosphate head, the phosphate faces the water, or kind of the inside and the outside of the cell. It's hydrophilic, which means it's water-loving. You can think of the suffix philic or phile as in, like an audiophile. An audiophile is somebody who's really, really into listening to music in high definition. The lipids, so these tails, the lipids face each other, or they face inside the membrane, not inside or outside, okay? So the lipids face each other and are hydrophobic. They're water-fearing. So what we would have is we would have another phosphate head here, and we would have a lipid tail. And then we would have water on either side. Let's just go down to the bottom of the page. We've got a picture here of the phospholipid bilayer. So here we've got our phospholipid bilayer, bi meaning one layer, another layer, right? So we've got two layers, and we've got the external environment or outside the cell and the internal environment or inside the cell. So you see how the lipids, they face each other because they hate water. We've got water on both sides, and so these phosphate heads, they face water because they're charged. They're really hydrophilic. Okay. The membrane is a complex combination of shifting components. Currently, this model is called the fluid mosaic model. What that means, it's fluid because parts of this plasma membrane, they can move around. If we go back to this model that we were talking about earlier, these phospholipids, they can trade places with each other. They can move around each other. This cholesterol unit, it can swap sides, and it can move around. These proteins can move around as well. And I'll give you a link to a video to watch here in a little bit after we kind of explain this a little bit more thoroughly. So go back up. So this is called a fluid because things can move around quite easily, and it's a mosaic. And a mosaic is just it's just a combination of a bunch of different things that make up a larger piece. Okay, so the mosaic is made up of a bunch of different things that we'll talk about here in a second. The components that can be found moving within the phospholipid bilayer include proteins and lipids. There are several types of protein in the fluid mosaic model. Some have sugar chains attached to them, and we'll draw that a little bit, and are called glycoproteins. Glyco just means sugar. So these are sugar proteins or glycoproteins. Now, these glycoproteins, they help aid in cell-to-cell recognition and signaling. So if two cells bump up against each other, cells don't have eyes. There's no way for them to be able to see what kind of cell it's next to. But when they have little things sticking off of each other, those things can interact with each other. And when they interact in the right way, then they'll know, oh, this is the thing I'm beside. These glycoproteins also help stabilize the membrane, and we'll talk about that in a little bit. So those are glycoproteins. We can also talk about integral proteins. Integral proteins sit within the phospholipid bilayer, either wholly or partially. We've got a couple integral proteins here. Here's an integral protein. It's embedded into, or it's integrated into, this phospholipid bilayer. Here's another integral protein, but this integral protein happens to be called a transmembral protein. Okay, so proteins that penetrate all the way through the lipid bilayer are called transmembral. Trans meaning spanning all the way across, okay? If you think of the Trans-Canada Highway, it goes from one coast to the other. All transmembral proteins are integral, right? If it's transmembral, it has to be embedded into the phospholipid bilayer. But not all integral proteins are transmembral. So what that means is both of these, right? This is integral, and this is integral as well. So they're all integral, but only the one that spans all the way across can be considered transmembral. And that's terrible. You can't read that, but that's okay. These types of proteins control entry and exit of molecules through the membrane, which we will talk about in our next lesson. There's also peripheral proteins, and peripheral proteins sit on the surface of the lipid bilayer. They also help in signaling to other cells. There's also several types of lipids, the phospholipids that we've talked about already, cholesterol, which interrupts the close packing of the phospholipids and allows the membrane to stay flexible, fluid, and stable. So this cholesterol, I know you may have heard that cholesterol is really bad for you, and in certain cases it is, absolutely. But in a lot of cases, cholesterol is absolutely necessary for you in order to survive. If there was no cholesterol, your phospholipid bilayer of your membrane would be too rigid. It would be so rigid that it might break apart if you added too much stress to it. So the cholesterol is necessary in keeping the fluidity of this phospholipid bilayer. Okay, there's also glycolipids, which are fats and with sugars attached to them. They aid in cell-to-cell recognition and signaling, and they help stabilize the membrane. They're also receptors for hormones and neurotransmitters. So what we're going to do is we're going to skip to the next page, and we're going to draw ourselves another picture that's very similar to this, but we're going to define some things in the phospholipid bilayer. Here we are on the next page, and boy, aren't you lucky. Oh, boy, you are so lucky. You have Mr. Walton, who is an amazing artiste. And so we're going to go ahead and draw our best 3D. I don't know about that, but that's okay. We're going to go ahead and draw our best diagram summarizing the structure of the cell membrane. Now, what we're going to do is we're going to start off with some phospholipids. So we're going to draw some circles. And because it's a bilayer, we got to have two layers. And each one of these is going to have two lipid tails. And that's kind of defining the edge of our membrane. Here we go. So this membrane would wrap all the way around in one big circle, right? But we're just going to have to deal with it. We're going to go ahead and draw our first protein, and it's going to be a transmembral protein. So what does transmembral mean to you? Well, transmembral means to me that this protein, I'm going to go ahead and do my best to draw. That's terrible. Oh my goodness. Oh man. Oh boy. So what I'm trying to draw is basically like a cylinder with a hole through it, okay? Because this particular transmembral protein has the ability to let things pass through it, okay? And we're going to learn that in our cell transport lesson tomorrow. But this is our first protein. So this protein here it's integral because it's integrated into the phospholipid bilayer, but also it's transmembral. It's a transmembral protein. Okay, let's go ahead and keep going. So we've got our phospholipid bilayer is continuing, and we'll continue it further on the bottom than we will on the top. So we're going to get our phospholipid bilayer with 2 tails on every single phospholipid. How tedious is this? You're having fun. I'm having fun. We're just having a wicked good time. Whoo, there we go. Perfect. Let's go ahead and draw our second protein. Now, our second protein is going to be a protein. Is this integral? Yes, it's integral because it's integrated into the phospholipid bilayer. It's taking up space inside that bilayer. So this is an integral protein. Nice. And this thing can help with the signaling of between one cell and another cell. Okay. All right. No problem. Let's go ahead and continue our phospholipid bilayer. Phospholipid bilayer. Pew, pew, pew. Man, I feel bad for you guys. You got to listen to me while we draw this thing. This is fantastic. Pew, pew. 2 tails on each. Oh my goodness. Oh my goodness. Ugh. It's so hard. Here we go. Oh, I think that's 2. Oh my goodness. There we go. Perfect. Now, on on the bottom side here, on the bottom side, what we're going to draw is a peripheral protein. So this is a peripheral protein because it's on the surface. It's attached to the surface of the membrane. And what we're going to go ahead and draw on here is we're going to draw some circles attached to it. And we're going to call this whole thing is going to be a glycoprotein. There we go. Perfect. So that's our glycoprotein, and it has the ability, again, to aid in cell recognition, but also in intercellular communication as well. Perfect. And something that we're missing, we're just going to continue this a little bit more. I'm going to skip a couple spots. And in here, what we have is and I can never remember how to do this, how to draw this. So I just kind of draw a little squiggly. That thing is cholesterol. Perfect. And let's finish drawing those tails. Why did I draw more of these? Just means I have to draw more tails now. Goodness gracious. Okay. So this transmembral protein and this integral protein, this is also the transmembral is they're both integral proteins, okay? But remember this one is transmembral. I think that's pretty good. I'm pretty happy with that. Now, something to consider for this is that this membrane, this membrane, it will act differently under different conditions. So if things start to get too hot or too cold, your plasma membrane will get really fluid or a lot less fluid. So what we want to do, we just want to talk a little bit about the particle model of matter. And then I'm going to get you to watch a couple videos just to help support today's lesson. So the particle model of matter, and you should know this, man, you should know this. You should know this because we went over it already at the beginning of this year, and we went over it in grade 9 as well. And this is actually a grade 7 topic, but that's okay. Whatever, that's okay. So the particle model of matter, first one is all matter is made up of infinitely tiny particles. But the particles are different. Particles in different substances may be different in size and mass. The next point, the particles of matter are always moving or are in motion. Adding or taking away energy will affect the movement of the particles. The particles of matter are attracted to one another or they are bonded together. Particles always have space in between them. And the space is tiny, right? So they're small gaps in solids, right? And very large gaps in gases. The spaces may be occupied by particles of other substances. Perfect, no problem. So here, if I've got a solid, okay, this solid, the particles here are still vibrating around. They're still kind of shaking back and forth, but they're fairly rigidly stuck next to each other. In a liquid, these particles can swap places, a lot like our fluid mosaic model. And in the gas, there's huge differences in space between them, and they're free to kind of go which direction they care to go. So as we add more energy, we give more freedom to the movement here. Same thing in our fluid mosaic model of the cell membrane. So if we have a cell membrane in a hotter temperature, then this becomes more fluid. And if it's more fluid, you want less cholesterol. If it's colder, then this becomes more rigid. They pack tightly, more tight together, and you want more cholesterol. So you actually want a different amount of cholesterol depending on the conditions that you live in. That's pretty much it for today. I've linked to 2 videos in the description that I want you to watch just to kind of solidify your understanding of the way that this fluid mosaic model looks, okay, and how it kind of behaves and acts. So let me know if you have any questions. Otherwise, have a great day. I'll see you later.